It is my great pleasure today to introduce Dr. Christelle Baunez, who is in the CNRS in Marseille, in France. And thank you so much, Dr. Baunez, for being here today.
You're welcome. Hello, everyone.
Yeah, so we're going to talk a little bit today about at least starting with her recent paper by Diorla, I guess, et al. In psychopharmacology this year. It's called Peer Presence and Familiarity as Key Factors to Reduce Cocaine Intake in Both Rats and Humans, an effect mediated by subthalamic nucleus. So this is a really fascinating paper. You have a lot of stuff in this paper. I wonder if you could just take us through some of the major findings.
Yeah, so the initial question was asked to me by a social psychologist, asking me if I could be interested at looking at the influence of the direct presence of a peer on drug consumption in my rats. And I said, oh yeah, that's a really cool idea. So I looked in the literature and someone had already started to look at it in a experimental psychology way, not looking at the neurobiological substrates. So since I'm obsessed with the subthalamic nucleus, I actually told the social psychologist, I'm okay to do experiments, but we need to address also the influence, the possible influence of the subthalamic nucleus in it. And then, of course, asking such a very interesting question, the strength of the study would be also to have a translational approach and use also the same question, address it in human users. So that's what we've done in that study. So we first developed a study, experiment in rats. So we have one rat self-administering the drug. And it's a double chamber with two compartments separated by a grid. And on the other side of the grid, the first experiment was placing a rat, not taking the drug, but taking it on the same day as the one taking and the other one being the observer. And so we first started with this, and then after we had another experiment in which both animals could self-administer. And we were interested at looking the influence of also of the identity of the peer present. So we looked at the influence when the home cage partner is the one on the other side of the grid, so that the familiar condition, and the unknown or stranger condition where we place the rat not sharing the home cage, obviously, being the stranger. And we added another condition, having a stranger totally naive to cocaine. So that was not used in that experiment at all, had never taken any cocaine, so didn't have a clue of what the other guys were doing on the other side of the grid. So there are the different conditions we used. And for the human study, of course, we are not allowed in France to give drugs to human subjects. So we couldn't really do it in live. So we had questionnaires written by socio-epidemiologists, and they ask psychostimulant users. So they are both users of cocaine and Ritalin in Marseille or crack in Paris. We have a cultural difference there. And we asked them about their last episode of consumption, how much they took, with whom they were, the identity of the person, whether people they knew, whether in groups or in just in dyads, with only one single person, and whether it was a familiar person or an unknown person. So we couldn't have any answer regarding the last condition we used in rats, with condition having a stranger naive of cocaine, because most of the time drug users take drugs in a context where they are with peers also consuming. So that was the limit of the human study, not paralleling exactly what we did in rats. So now the main results, of course. Yeah, just to mention that, of course, in the rats, since I mentioned we wanted to look at the influence of STN, we had sham control animals, for all these conditions I described, and subthalamic nucleus excitotoxic lesion animals. So that was still lesions, because, you know, I always have this approach before developing very sophisticated studies using optogenetics or deep brain stimulation. I want to be sure that it makes sense to spend all this energy. So the first step is always to look at the effect of the lesion. And since the subthalamic nucleus is still considered quite homogeneous in terms of glutamatergic structure, it's not really useless to start first with an excitotoxic lesion. So I'm still very much a supporter of this approach.
Absolutely,
And,
there's nothing wrong with these classic methods, especially when you know exactly how they're working. And if there's no advantage to the fancier new method, my sense is, you know, it doesn't get you anything else other than a little bit of flashiness, perhaps literally in some cases. But nothing wrong with lesions. Totally with you.
Okay, so well, now briefly the main results are really striking, because I'm going to start with the human. And the reason why I start with the human, you will understand, because it's not necessarily what we would have expected. But what happens in human drug users is that the most consumption they report is when they are alone. And it decreases progressively. And the first condition of decrease is in direct presence of a peer they know. So that's the least decrease of consumption observed is when they are in dyad with a familiar peer. And the major decrease reported in consumption is the condition when they are in dyad with someone they don't know. So it's counterintuitive to my point of view, because I would have thought that that would be a stressful situation, and they may be tempted to increase their drug intake, but no, it's a decrease. So if you have, so when we discuss with people and we try to analyze why would they do that, immediately people will say, okay, it's a judgmental thing, and they fear to be judged badly, so they decrease their intake. Now, interestingly enough, thinking of this moral judgment, the interesting thing is that what happens in rats is that we find exactly the same results. So rats take the most drug in the condition when they are alone. And then progressively they decrease their intake in presence of a peer, but with a familiar peer is a small decrease that is enhanced by the lack of familiarity. And then in presence of a stranger rats, that's when they take the least. And even worse, even better for them, because they take very much less if they are in presence of a stranger peer naive of cocaine. So now,
does that imply that the naive sort of I don't know, square rat or you know, non-partying rat is like extra judgmental or something like that, or just confused, or do you have any sense?
I don't know. The thing is, first of all, I was surprised that they could even make a difference. So, but bear in mind that we only separate them with a grid, therefore allowing them to sniff each other and touch each other through the grid. So they have social interaction, so they can exchange in many ways. So we will go back to the vocal communication between them, of course, but I think the smell, and we all know that an individual taking cocaine have their urine smelling. So if they are totally naive of the drug, probably the smell is different, and maybe that sends a signal. I don't know. I don't know how to interpret that, honestly. Really? But that's quite interesting, because what we wrote in the manuscript, and maybe some editors didn't like that, but we were in France in the middle of a debate regarding the consuming rooms, where addicts could go and inject in a safe place where medical care people could be there. So looking at the influence of the direct presence of strangers, possibly non-users, is quite efficient at reducing the intake in rats. So maybe for the human being, it would be interesting to support these consuming rooms. So that was the take-home message we wrote. So now I mentioned the fact that in rats, we had two conditions. And so what I described was a result when the peer on the other side of the grid was just doing nothing but watching what was going on. But we, interestingly enough, when they are both self-administering, there's no facilitation of the behavior, but they actually both decrease their drug intake. So it's as if they were having a self-control on their drug intake. So that was really puzzling, actually.
Really interesting. So you're saying that even two rats that are concurrently using drug will suppress, just the mere fact that they're near each other will suppress each other's intake.
Yeah, yeah.
Wow.
So that's what we've observed. But Smith, the psychologist I referred to, who had initiated this type of research in the US, only doing behavior, was actually had published a paper in which he showed that it was the opposite, but they were learning the acquisition of the self-administration behavior together. While in our case, they both rats learned individually, alone in their experimental room. So they learned the self-administration behavior being alone. So, as we report, being in the condition in which they take the most. And then after, they don't regulate their intake if they are placed in presence. So that was a different setting that may explain the opposite effect. But surprisingly enough, when we're currently running experiment with alcohol, and we don't find the same thing, because alcohol is really definitively a pro-social drug, and it tends to facilitate the intake to have the social presence.
Oh wow, that's really interesting. I was about to ask you about other drugs. That's just, it reminds me of when I years ago was a master's student in Harriet DeWitt's lab doing human research along these lines. We did studies with various drugs, but with alcohol, it was the only way to do a consumption study in humans is if there's other humans around. It's a confound, and sometimes they don't like each other, and sometimes they're like different people on the two sessions and stuff. So there's downsides, but people don't drink by themselves in that setting. Maybe alcoholics do, but these weren't alcoholics. So fascinating finding. And I just love the fact that you have, you've done a very ambitious thing in this paper, coming up with experiments in both rats and humans that are obviously not exactly parallel, but really have a lot of common themes to them, and really make a very similar case across species.
Yeah, it was really exciting to have the parallel between the two species, and I was really influenced in that by the work of Aldo Badiani as well, who had looked at the context in which rats, like human, will prefer opiates in their home cage facilities, while they would take psychostimulants in another setting when we take them to the experimental room. And I loved that paper where they showed that in parallel in rats and humans. So I was really much influenced by that work that I find fascinating. So, but it's difficult to publish this type of work, especially as I was telling you before, that the fact that we use social science to work on the human side and basic experimental work in rats is difficult to find editors who dare to confront both approaches. So that was really a big struggle. But Elodie Giorla was the first author. That was her PhD thesis, and when she's put a lot of effort in doing all these experiments, and that was a good reward. We finally got it published.
Absolutely, and I'm glad you did in psychopharmacology as well. I mean, there's not
exactly
that many journals that, I mean, this is a largely behavioral study, especially obviously in humans, and it's kind of an unusual thing, but it focuses on behavior, and the parallels are clear. And I think that's kind of a perfect journal for it in that regard. the emphasis on behavior and whatnot in psychopharmacology. Another question I had about your study is with regard to sex. So I noticed that this study was in males, and the sort of the partner was male, I presume, also in the case of the rats. And I didn't notice whether you had males and females in the human population. I'm just curious if you could comment on the interactions that sex may play in these.
Of course, fantastic question. I mean, this paper is actually the first work we initiated with this question of the proximal social factors influence on drug intake. But of course, it's the beginning of a new big line of research that I'm developing in my lab nowadays. And there are so many questions I would like to address. And for example, the influence of putting a female on the other side of the grid. Are they going to play the tough boys and taking more drugs, or maybe less? Who knows how they can interpret that? So that could be really fun to do that. So we had many projects like that. And of course, we are also aware that nowadays it's important to plan experiments always mixing genders. So we're of course, like everyone nowadays, when we apply for grants, we also put the gender issue, and we plan to do that also in female, but that's in the future
Sure,
papers.
Yeah, there's always more interesting questions, and there is time to ask them. But I'm really glad to hear that you're following that up. I think that'll be a fascinating set of results as well.
Oh, definitely. Yeah.
And then I also wanted to ask you about the subthalamic nucleus. You mentioned before that you've been studying this for some time, and I'm curious if you could speak about, maybe a little bit about the results you found here with the lesions that you studied, and then also perhaps more broadly about the potential therapeutic benefits of trying to target STN in some way in humans for addiction.
Yeah, so in that study in particular, the STN lesion has an important impact in the term that it abolishes the differences obtained between the familiar and the stranger peer. So as soon as you place another individual, whatever the identity, the STN lesion guys show an immediate reduction of their drug intake. And it's very interesting and question the influence of STN in social recognition. So that's going to be the topic of a next paper coming, being submitted soon. So published maybe in a few years' time. But that we have looked at that question. But interestingly enough, we also looked at the effect of STN deep brain stimulation on many aspects of addiction. And that's my work in the last 15, 20 years. And the first striking observation with STN is that when we did an inactivation of the subthalamic nucleus, I don't know if people are familiar with the structure of the basal ganglia, but this structure is a small nucleus, glutamatergic, and being overactive in Parkinson's disease. So it's currently the target of the treatment for Parkinsonian patients. And very efficiently, we can implant deep brain stimulation electrodes in the subthalamic nucleus in PD patients, and they recover. Most patients operated recover their motor functions. And then when I studied the effect of STN inactivation in the context of Parkinson's disease, that's how I was puzzled by some results on motivation and suspected there could be something going on. So checking what was going on, I looked at motivation for sweet food, and I observed an increased motivation for sweet food when you knock down the STN, either by lesioning, inactivating with optogenetics, or with electric deep brain stimulation. But then I was really wondering whether we could have an increased motivation for everything. If we follow the textbook on motivation, if you find an increased motivation for something, you expect motivation to increase for everything. So I thought, these patients, they take dopaminergic drugs for their pathology. So they may become addicted. So we should be careful about that. So that's how I came to look at the influence of STN lesion on cocaine self-administration. And that was the paper we published in 2005. So you see, that's a long time ago. And where we found an opposite effect on motivation for cocaine. In contrast with the increased motivation with sweet food, we had a decreased motivation for cocaine. So that was positioning the STN at a very interesting place in the reward circuit, because that's where you find an opposite effect depending on the reward on board. That is really totally, that wasn't known at that time. And we couldn't explain it actually. But second interesting aspect, not only because it was interesting intellectually speaking to think of how that could be true, it was also that the result was exactly what we want to do with addicts: reduce their motivation to take the drug without decreasing their motivation for everything. So I thought, wow, we have a target for addiction. But now we need to demonstrate, because people were like, oh, Baunez, what is she coming to do in the addiction field? So, so I really needed to look at the different criteria of addiction that we can model in rats and prove that it could be actually a very interesting target. And we have the tool, because we know that we don't need to do lesion in the brain of an addict. We can implant electrodes and apply deep brain stimulation. So we've done that, looking at the influence on the loss of control over drug intake in the escalation model of Serge Ahmed and George Koob. So, and we could see that we can have a preventive effect, and we can block escalation by applying deep brain stimulation or making a lesion of STN. And if once they have escalated, we can prevent a re-escalation after a withdrawal period. And we recently published in PNAS in 2021 the influence of STN stimulation on the compulsive seeking of the drug. So our rats resistant to a punishment associated with the drug seeking can be reduced by stimulation, but now it's at a different parameter of the deep brain stimulation. So the story always gets more complicated. Of course, it would be too simple to say 130 Hz works for everything. No, indeed, not on all criteria, the 130 Hz is efficient. So we may need to move towards closed loop system recording, because we have also a signature of abnormal oscillations in the STN when the animals lose the control over the drug intake. And in this process, some type of signature in the STN can be predictive of which individual is going to be resistant to punishment. In other words, vulnerable to addiction. So now we start to have a nice story. I'm writing up now a paper using macaques to show that deep brain stimulation of STN can indeed reduce motivation for cocaine, and while increasing motivation for applesauce. So we really show this, that our hypothesis is quite good. Now,
yeah, that's so interesting.
Now, before translation to humans, it requires a few more steps. But interestingly enough, to go back to the social questions, the recent paper we're finishing now is showing that deep brain stimulation of STN applied in rats having lost their control over cocaine intake can be, so they can go back to a recreational consumption with deep brain stimulation, or with simply the presence of a stranger peer. We observe the same reduction. And if we combine both, sorry, it was not deep brain stimulation, it was optogenetic inhibition of STN. And so if we combine both, we actually don't potentiate. So that means possibly the influence of the presence of a stranger peer at reducing the drug intake is mediated via the STN network.
Wow, so interesting. And so do you happen to know if any of the people that have these DBS electrodes for Parkinson's or whatever, do they report social changes, recognition in particular, or anything else?
That's an excellent question. I keep asking my colleagues, neurologists, and they haven't looked at it really. So it's not something that spontaneously the patients report, but interestingly enough, we have another very interesting observation in clinics is that some Parkinsonian patients become addicted to their levodopa treatment. So it's called the dopamine dysregulation syndrome. And some patients suffering from this syndrome, if they have ST and DBS, they stop seeking for the L-DOPA and trafficking their prescriptions to take 10 times the dose they should take, which is usually what they do when they suffer from this syndrome. But it can, ST and DBS can be beneficial on these patients. It was only, the DDS was only discovered in the early 2000s. And of course, patients were not spontaneously telling their neurologist, oh, by the way, I'm trafficking the electrodes, the prescriptions because I want to take L-DOPA so much. So it needed to be asked in a certain way. So I suppose that the social recognition possible changes may be hidden or ignored, and they could be investigated. That would be really interesting to look at that.
Absolutely, and it really just goes to show the value of doing this kind of reverse translation or bidirectional translational types of studies cross species.
Yeah.
Because, you know, there's probably all kinds of stuff that's happening in humans with these experimental electrodes that nobody just thought to ask them. And neurologists aren't exactly trained to ask these kinds of questions about psychological changes and things like that. So this work is, I think, really just extremely important for that reason and many others. We should probably wrap up, but just wanted to ask if you had any other comments on what are the really important next steps, other than obviously some of the stuff you've talked about in your own research program, but in terms of like implementing new treatments, maybe to do with STN, or in terms of you know, the most exciting directions you see in the next few years in the field.
Definitely the social context is really something that is blooming, and we also think of the work by Marco Veniro with Yavin Sharam and Luc van der Schuren with a social play as an alternative reward. So all these strategies looking at alternative rewards, also I'm thinking of the work by Serge Ahmed, of course, and so many people are looking at diverting the interest for the drug towards something else. And so I think that's really an interesting direction. And to also to go back to some of the studies we've done and how they can translate to human, we've looked at how the rats' presence can influence the drug intake, and can we replace the social presence by just playing a playback of ultrasonic vocalization? And we have shown that we can induce a reduction of the drug intake by displaying positive ultrasonic vocalization, not negative ones. So the positive message is beneficial at reducing the drug intake. And in all arm policies, we always focus on negative messages. So maybe, again, a reversal translational work can lead us to try send positive messages to drug users, and maybe we will help them better.
Fascinating. I think that's just a fantastic idea, and this work is just extremely thought-provoking. I really appreciate you telling us about it today. So thank you again very much for your time,
Thank you,
Baunez, and I look forward to seeing what you're going to be up to next.
Yes, thanks a lot for your invitation. That was fun.